Today our second lecture, Tumor Biology, and we will speak about tumor, about how it happens, which mutation are reason of tumor. And I let me introduce. My name is Елизавета Ageva. I am head of department medical biology, and I will teach you in our practical class. And before our lecture, I want to say you how we will have our practical class. Who was in big date know that we was work with 3 modules, and every modules we make report, and this is report. It was for mark, for your marks, and give check of full discipline. In today our course Tumor Biology, Cell Biology, we will have same direction, and we will have 8 class, 7 practical class, and last class will it is mini conference which you will give your own report. And we will work in small group. Every group will 3 persons, and total will 5 subgroup in one group, in one class, and you will make 5 report. Every class from 1st till 7, we will step by step prepare our final report. Our topic will about transcription and translation, how it happens in normal healthy cells. In second class, we will speak about tumor of biology, today topic. Which mutation, how it make normal cells transfer to tumor cells. The second class will about mutation. Mutation in which genes need to happens for this transfer. We will speak about proto-oncogene, anti-oncogene, about drive and passenger mutations, about transcription factors, about different waves of cells which make damage and which make normal cells transfer to tumor cells. In fourth class, we will speak about transcriptional factors. 5, it is about signal, opposite. 5 signaling way, and 6 will about transcriptional. It is topic of next lecture. Then we will speak about target therapy, because it is head of today modern medicine, about therapy strategy, and we will need to understand how it is work. We not will speak about pharmacology, about pathology. We will speak in our course about biology. Biology gene target, it is main of our course. And it will give you information and in future can help you to understanding all pathology and pharmacology questions. And I said in second class you will divide it for your small subgroup, and you will take information from article, from database, how it was in big data, and you will use your previously knowledge from biology about transcription, translation, about mitosis, meiosis, about chromosome structure, about classification of mutation, gene mutation, chromosome mutation. And you will need remember all topic which you take in big data in medicine and biology and practical genetic classes. And of course, I think all our teacher will help you to understanding. And of course, I hope that you will give answer to us and will it give for us many possibility to make our course better and better. And we need to start from basic terms and concepts. Tumor is a process represented by a newly formed tissue in which changes in the genetic apparatus of cells occur to disrupt the regulation of their growth and differentiation. Cancerogenesis. It is a process of tumor formation. Apoptosis is a programmed death cell. Angiogenesis, the formation of a vascular or blood vessels. And we need to say about all tumors may be malignant and unmalignant. And if you can to look of my picture on this slide, you can to look first picture have a round of tumor, and here separate from any tissue, and if it will have cover, it will unmalignant. He only press for tissue which near this is tumor, but opposite Malignant tumor, it is tumor which can, as cancer, make growth in tissue as finger for different tissue, different cells, not have separative shape, and will change normal and abnormal tissue and cells. Also, in this picture, you can to see vessels which grows to tumor and give a special possibility for this tumor to take vitamins, hormones, growth factor, carbohydrate, and other what need to grow very fast of cells. Cancer, it is a group of disease which have in a reason mutations, genetic mutation or chromosomal mutation. And if you will look on my slide picture, which can to see normal and cancer cells, you can to look. Normal cells have all genes, all DNA normal. But in cancer, you can to see mutation and only part of DNA, which give many new activity for bad genes and for tumor development. All tumors has uh, properties, and first it is immortality. Immortality. It is a problem which happens from apoptosis. When program of apoptosis, program cell does not work, and not happens control of cell growth regulations. Second, it is loss of growth control or inhibition, special inhibition of this process. Impossibility of terminal differentiation. It means that all cells of tumor cells not have tissue speciality. They look as previously cells. Uh, Example in myocard, in muscles, in brain, in glandula, in skin, all cells not will look as these cells. They will look as previously primitive cells and not have um, look of differentiation. Next, it is ability to invasive. It means all cells of tumor can make growth between normal cells, make moving normal cells and take this space for his own growing. Next, it is cell separation. Usually, all cells take together because connection between cells give to community of cells and give all common structure. But tumor cells can disconnection, separative. It is give possibility to invasive growth first. Second, it is give possibility for metastasis because one cells can go to blood vessels and with blood go to another organs. And one more important properties of tumor cells, it is angiogenesis. It is growth of vessel and blood connection for tumor cells can take growth factor. You can to look of this slide, and here you can to see two type of cells. Normal cells, it is number 1, and apoptotic cells, it is number 2. And main is this is apoptotic cells that all information genetic information in cells and all organelles not will come out from cells. They will keep in cells, make small size restriction of genetic materials, and will go to phagocytic and keep in organism without any damage for outside. We will speak about way of cancerogenesis have many theory, but we not will say about all theory. You will speak about this in next course and next discipline. Now we will need speak only how it happens, one theory, and this is theory or a model of stochastic generation. And in these pictures you can to see at left part cell with mutation. You can to see some of cells and one cells with mutation, but they have look as normal. As normal. In future, it will give many new cells, hyperplasia, and here we can to see that they look same as normal cells but have a mutation. Genetic new, phenotypic old. Then in this story, when many hyperplasia and many new cells uh, can to happens new mutation, and it give a new cell story. This is blue. It will new clone of cells, and it will give again new growth, many cells, and now cancer in beauty look as tumor, and now you can to see three cell population with different color, uh, violet, blue, and orange. And it means here present many different genetic clones, genetic mutation, which happens always in tumor cells, and in result it give a big tumor which will growing in tissue, invasive cancer, and will give some of cells in vessel blood, which will go with blood and make metastasis. Tumor have many type of classification. We will speak about some of them because we remember that we study biology and we will need to understand biology of tumor. And first type of classification which I can to say to tell you, it is sporadic cancer. It is most common form. It will meet in human in 75 in 80%. They have multifactorial story, and it is have location position in body. Example, mutation come in gaster, and only cells of this tumor will have mutation. All other tissue and cells not will have any mutation, will normal. And this is mutation will work only in this story with gaster and not will give to in future same mutation in child of this person. Opposite type of cancer, it is family. It means we speak about monogenic or recessive or dominant. More often it will dominant. It is as Mendel rules when one mutation give one disease. In this case, it will tumor disease, and it means every cells will have this mutation in one body, and it means this mutation can to be in future and in parents, in future child of this person and parents of this persons. And more seldom we can speak about hereditary tumor syndrome. It means it is a mutation which will have in family story, but it is not tumor. It is not reason of tumor. It is only predisposition when any factor can to lead tumor, but if we make this includes this factor, so tumor not will come. Next classification, maybe you can to meet this is classification. If not, you will teach this is classification in your next pathology, pathophysiology, and so on. But I only want that you listen and understand how it works. It means carcinoma, it means sarcoma, carcinosarcoma, melanoma, tumor of lymphoid or hematopoietic tissue, and blastoma. And for easy understanding, you need to remember it is from which tissue it happens. So we speak about carcinoma, sarcoma, and so on. Reason of tumor, it is not only mutation, not only genetic factors which we can to find in chromosome or in genes. It is also maybe epigenetic cell damage, and it will again about genetic materials, only it not will in future and new generation. As example, we will understand if we speak about mutations in genome, we will speak about mutation in next genes, proto-oncogenes, suppressor genes, and DNA repair genes. Also, we need to understand and make differences between driver mutation and passenger mutation. And you need to understand that driver, it is provide uncontrolled growth. It is main mutation which can to lead to tumor. And passenger mutation, it is a mutation which happens, but it not maybe very important for cancerogenesis, for oncogenesis. And about proto-oncogenes, suppressor of genes, and DNA repair genes, I will speak later. But about epigenetic disorders, as example, impairment in gene expression, methylation, histone acetylation, chromatin rearrangement, splicing, and microRNA. We were speaking in our previously class and will repeat again in future. And now a little explain about proto-oncogenesis. This is normal genes that regulate cell proliferation and differentiation. They serve as a precursor for oncogenesis. And if you will look of my pictures, you can to see proto-oncogenesis, proto-oncogene not work in normal chromosomes. And not work any one time. It is left side, left way, and it give normal cell growth. But if in this gene, proto-oncogene, will happens mutation, this is proto-oncogene will become oncogene, and it will give loss of growth control, and tumor will happens. Example of proto-oncogenes, it is normal genes which will work more expressive in new tissue, in new cells, and will give tumor phenotype. This is a receptor of tyrosine kinase and change which happens after mutation. It is gene amplification and overexpression, and this is normal growth factor. If growth factor will give information for receptor, will work many more than in normal situation, and so all cells will divided. Cell proliferation will high level, and will happens many new cells. More famous gene mutation happens in gene K race, N race, H race. This is protein function. This participates in the transmission of mitogenetic signals and the regulation of morphogenetic reaction. And it is more distribution in many type of tumor, and the match of these genes will to give for new cells possibility for divided and divided. Next, it is BCL second. It is anti-apoptotic proteins which not active or not work in normal cells. Uh, if he will activation, they will make stop apoptosis, and it is a way which can use example viruses or some of drugs, and also not good. But it is will make stop of cell death. But in normal cells, I will repeat, in normal cells, BCL second not work or little work. When will happens mutation in this gene, they will work very high level and very strong, and not will give cell death happens. So tumor cells will without any type of death and will divide it and divide it. Now, also next genes you can to look in this slide, and also this is genes which work and have any function, but in result of mutation will activation or amplificated of these genes, and they will lead to many problems for this is body and for these cells. Here you can to see mechanisms of oncogene activation. Look and then I will explain. And here you need to see some type of mm, some type of waves of activated amplified proteins production. Example, here we can to see translocation or transposition, and here in one gene we can to look two part of genes. More blue, it is structure information about protein, and light blue new promoter, no, in this new promoter. Light type, it is promoter or regulation part of genes which can regulate it, how many speed, how many levels, how much need to expression this is proteins. And this is first situation when we can to look structure part of gene from one type of gene, blue, and light blue, it is a part from another part of gene promoter. Example, if will insertion or a translocation, and one part of genes will connection with another part of another genes, and it will give information from one promoter which will make many expressive of structure information in other genes. Next happens gene amplification, and regulatory genetic sequence will make 2, 3, 4, 5, 6 times, and it also will give information of many transcription and translation and synthesizes of proteins. Next, we can to look that promoter or control element will new. It is will with mutation and will have new possibility, new information, will give for transcription of genes. And next part again will point of mutation or maybe CNP, which we speak about previously our discipline. That in will give new information in amino acid LEDs of genes. Next, we will speak about suppressor genes. It is genes whose inactivation increases of development tumors or anti-oncogenesis. And here we can to look again same story about normal chromosomes and normal cell growth, but also maybe situation after mutation. And you can to look mutated tumor suppressor genes, which one chromosomes have mutation, second not have. If you remember, all chromosomes have duplicated, have pair from mother from father. And if a mutation happens only in one chromosomes, it means they not will give any new information because normal genes will work as dominant allele and will suppress this is mutation allele, mutation genes. But if mutation again will come and will give problem for second normal chromosomes, so all these genes will work as damage and will give bad tumor information for all cells, and tumor will come and loss of growth and will happens loss of growth control. And example of suppressor genes, an example, it is P50 53 protein. It is a transcription factors. Also, it works as regulators of cell cycle and apoptosis. And it is no, if it is transcriptional factors, it means make control of many gene expression. Here, cadherin, it is also protein which take part in intracellular interaction, and it will a part of signaling activation different transcription factors. We will speak about little later. Also, it is adenomatosis polyposis colon protein, and mutation in this protein will also make colon tumor. It is VT, it is transcription factors, which also give many problem for happens disease. Now we will make a small break time for 5 minutes, and then we will continue our lecture. And we continues our lecture. And next, you can to look example of tumor suppressor gene is P53, and you can look how make many differences in one cells. This is protein and how they work, how many function, apoptosis, DNA repair, synthesis, differentiation, cell cycle arrest, and how many factors can to change these proteins. Example, it is nucleotide deletion, radiation, oncogenesis, DNA damage, telomere shortening, head shock, metabolic change, hypoxia. And here I need to say you about more famous tumor cells culture. It is cells lines of cell, HeLa. It is very famous. This is story start from woman. You can to look of these pictures. Henrietta Lacks and her two letters from name and two letters for forename will make together and start a name of cell lines HeLa. And he was diseased with tumor in 1950 first years. Then she will died. She was died. No, she died, and her cells was cultivation and make line, and now it is as a standard for research of tumor cells. And the next importance which you need to know, it is two-hit theory of Hudson. Alfred Hudson in 1971 give this is theory and explain. He speak about need two minimum two mutation which will give oncogenesis. What if we return, you can to understand. Example, one mutation was happens in one um, chromosomes, then what was happen second mutation in second chromosomes. But in real, it need to happen 6, 5 different mutation in different chromosomes and will give different initiation of different genes for tumor start. And this slide show to us how it works. And you can to look that was one cells, they was divided, but in one times was happens mutation, and then some of times normal cells was divided and tumor cells also divided, and then many tumor cells atypical or new phenotypic, not normal, abnormal phenotypic was make tumor, big tumor. Some of them can separative, come to vessels and go to next organs. It will, we will call this is a metastasis process. And also we need to understand that this is cells, it is target cells which happens, which was sensitive for mutation, and they have target genes which we can no, reparative, regenerative, which with use the drug, and it will give many healthy for persons. Need to remember or need to conclusion main position. It is oncogenesis. It is DNA repair. genes, and it is a tumor suppressor genes. It molecular mechanism of cancerogenesis. If we speak about stages of cancerogenesis, we will need to speak about initiation, promotion, progression, and metastasis. And in every step, we will speak about genetic mutation, which will work as target. And first, it is cancer gene causes mutation in proto-oncogenesis and anti-oncogenesis. Proto-oncogenesis are activated and become oncogenesis. Anti-oncogenesis are suppressed and also became oncogenesis. A cell still with an unchanged phenotype is a precancer. It is characterized by a violation of the control of important function, cell growth, division and differentiation, or latent period. As example of gene mutation, it will cell proliferation regulators, example family RES. It is transcription factors, example family MIC, and a cell cycle regulator, it is P53. It is apoptosis family, it is example protein BCL2, and a gene of reparation systems. Next stage, it is promotion stage. And here we need to say about clonal expansion of initial cells. For it will works, need make action of any stimul, and this is stimul will call as a promoters. The promoter causes an increases in the proliferation of dominant tumor cell, immature cell of the original tissue. Cell acquire a tumor phenotypic. Now it is we speak about genetic tumor phenotypic. Genetic tumor and phenotypic tumor. Next, it is promotion stage, and also we speak about stimulation of growth in precancerous cells occurs under the influence of growth factors, hormones, characterized by telomerase dysfunction, defect in chromosome segregation, dysregulation of the cell cycle, and DNA repair, inability to implement of apoptosis. And also here I need to take your attention about telomere dysfunction. If you remember from biology discipline, we speak about telomerase. It is enzyme which make all time telomere repair. Telomere, it is end part of chromosomes which will divide it every times when will chromosomes divided. And this is structure have end of your life story near 40-50 cell division divided. And telomerase need to repair all times for make new generation. But this is enzyme not will work always excellent. They work in young, in childhood, and then your activity down, down, down. And this is a story basis as theory of old And many researchermen want to find how make telomerase always activity for make longevity of persons, of people. But opposite side, it is tumor when telomerase always very high activity and give always telomere repair, and it give for cells all times divided, divided, and divided. Next progression stage, cell have changed genotype and phenotype. They divided indefinitely. The process of destruction, necrosis, inflammation in the tumor itself led to the appearance of cell with different mutation. The genotype of the tumor in the monoclonal stage is strongly different from that in the polyclonal stage. In this stage, many genotypic cell variations are present in the tumor, which complicated therapeutic option. And here we need to remember about type of mutation. We know about mutation may be gene level. It may be nucleotide, single nucleotide changes. It may be insertion and deletion. It may be amplification, tandem repeat, tandem gene repeat. Uh, Next level, it is chromosomal mutation. And here we will speak about change of structure. Example I said of translocation when we will speak about HEMER genes, structure part of genes from one chromosomes will connection with promoter part of another genes from another chromosomes, translocation. Or we can to say about loss of part. Example, will deletion. No, here, one chromosomes has mutation, red part. Second chromosomes normal. But if mutation will give deletion of this part, so when aneuploidia in one chromosomes and second mutation will with second chromosome will with mutation and without any par, so not suppress and will give disease. It will genomic mutation, genomic type of mutation. And genomic mutation, example, when we speak about not aneuploidia, when we speak about trisomia, also additional dose of genes will give tumor genesis. And also we need to remember about epigenetic remodeling, DNA methylation, histone modification, and change in microRNA profile. It means small structure which can block or activate translation on ribosomes. In our class, we will speak about genetic instability. It is, we will speak about dynamic process when in tumor, when many different genetic clone, maybe always concurration, maybe fight between different tumor cells for blood, for hormones, for vitamins, and damage each other, and all times make new generation of mutation. And it will make a way of different mutation, and it will genetic instability. Next type, it is chromosome instability. It is also when chromosomes will all times break and damage and lose part of chromosomes, and also some of genes which was before in suppressed condition will make work as dominant. We will speak about macrosatellite instability, which happens different condition with different genes, and they will work as amplified, different work as um, synonymical and non-synonymical mutation, which led to change amino acids. We will speak about laws of heterozygote. I said about this when show you example. This example, which two mother and father chromosome, one has mutation, second normal dominant, suppress this mutation, and if will chromosome normal of this pair lose her part, mutation part will work as alone and give oncogenesis. Which cause led to mutation and led to oncogenesis. It is chronic injury and inflammation, because in induration and inflammation always will death of cells, and place of died cells need make new cells, new generation, and if will mutation, so new generation will divided and will tumor. It may be viruses, example hepatitis C, Epstein-Barr, human papilloma virus, and bacteria Helicobacter pylori. They can to lead against in many regulation function of cells, example block mitosis, block apoptosis. It make example viruses, restriction of DNA human, take his information, and then this is cells will work to amplificated viruses genes, but in result will many mistake of cells, and it also will give any mutation, any way for oncogenesis. It will different transcription factors which happens with smoking, with alcohol, with drug, and many environmental factors. It will factors same as translation. Example, alternative splicing, which led new variation and give not excellent variation, which will make tumor genesis, and also it will post-translation modification after translation when protein will happens, and it will also atypical new formation, which we will speak about onca protein. And also I need to say you about you will work with first ethical. Every teacher will give to you ethical for your group in for your work in group, small group, and also you will find information, and also you will use base date which we have in internet space, and some of them you can look here. It is the Cancer Genomic Atlas program, it is Human Cancer Model, initiative catalog. It is genomic data commons and structure and functional genomic data portals. And I hope our work will successful, and we will meet with you in our class and next lecture. Goodbye.